and they're going to start attacking. This is the key behind the vaccination process. Here are several days where your body is infected and the virus is spreading and its numbers are increasing and you are unable to do anything at that time. So you're getting an infection throughout the body. It is spreading quickly. But if you've been vaccinated, you've already gone through this time period. So now when you get exposed, you already have your soldiers in place. Does that make sense? You're not having to wait and train them and tell them what to do and get them ready to go. They go. And so this is the difference. We get an instant response. That's the key. Now that instant response sometimes is enough to fight all of the viral particles coming in. Remember my Lego analogy. If we get too many viral particles, then unfortunately there's not too much I can do, right? I have to wait while I get those viral particle numbers down. So remember, a vaccine is a tool that helps us. It does not guarantee that you're not going to ever get sick. It's not a guarantee. Okay, any questions on this graph? Isn't that fascinating? These B cells, these memory cells, they stay in your body for decades. Like I said, it's different levels depending on what the organism is and the size that they are as to how long they'll stay. Rachel.
So that's why they say it takes about 2 weeks for the actual vaccination to start taking effect, because at that 2-week mark,
you got it.
Days, the memory cells are.
You got it. This is also why a lot of times people will tell me, oh, I got the flu vaccine and then I got the flu from it. No, here's what happened. They got the flu vaccine, and when do you usually get the flu vaccine? Do we get the flu vaccine when flu isn't circulating or when it is present? When it is present. So we get our flu vaccine when it's already present. So we get our vaccine, and then we get exposed to the flu at around the same time. Maybe even at the pharmacy where we went and got the vaccine or whatever we were doing that day. So our vaccination process is going to take 5 to 7 days, but flu has an incubation time of 48 hours. So if I get exposed during that time period, it's going to feel like the vaccination caused it. The vaccination is a dead virus. You're not going to get the zombie flu, okay? It's not going to be like the virus is going to come back. No, you got exposed to a live virus somewhere, okay? So the same is true also with chickenpox. Sometimes you'll hear that children are getting chickenpox vaccines, and it's a sequence. They get one shot, and then 6 months later they get the other. And during that 6 months, they get exposed to somebody with chickenpox. And so they get sick. And the parents are like, well, why? They were vaccinated. They had one shot. They didn't have enough time to build up enough antibodies. So yeah, so there's a lot of science behind the vaccination process, which a lot of people don't understand, that leads to this misunderstanding. It's like, oh, they don't work. No, they work. They just need time. Got to give them time. But we're into instant gratification. Okay, so how does the B cell know what antibody to produce? Well, it turns out they really don't. They just throw a whole bunch of antibodies out there. That's really what they're doing. And then depending on which ones work, that's the one that we make a whole bunch of clones of. So with this then, it's really amazing because we're exposed to millions of antigens throughout our lifetime. There's been some estimates of up to 10 million antigens. That's a huge number. But if we think about our genes, our genetics only have variable genes up to 25 to 30,000 genes. So how do we get millions from thousands? Well, it turns out that our antibodies are really unusual. They have this random variation in their DNA. So we learn in biology that all of our cells have the exact same DNA. That's what we're all taught. It turns out that antibodies are an exception to this rule. They have the same DNA, but they have a bunch of what are called variable regions. And these variable regions are unique to the B cells. And these variable regions let them differ from every other cell in the body. And what the variable regions do is they take a sequence of genes and they card shuffle them, basically. They have what are called transposons. They shuffle where the bases are. And because I'm shuffling, it's like taking 30,000 cards and shuffling them in these areas. So it gives me a lot of variation, which is how I can fight millions of antigens. So it's kind of a cool process. So if we look at this then, the variable chains on a B cell are going to be mostly seen in those light regions. So the purple colors are the ones that are going to be variable. The solid colors, the constant regions, are the ones that are going to be staying the same. And here's an example of how their process works. So the B cells will have a sequence of genes, just like all of our cells do. But in their variable region, they'll have special enzymes that they produce, so the only ones that produce them, that will separate the genes and reshuffle them together. So I can cut, rejoin, and I make something new. And I do this constantly, so I'm always making new B cells. Yeah.
So are these genes present in every cell but only activated in these B cells?
So these transposon areas, they're present in a higher number in the B cells. So that's a good question. We all have some variable regions in our cells. And that's one of the things that they're looking at for genetic work right now. That's one of the areas that they're really studying. We all have a few variable regions. B cells have the most and the highest number.
And is that true of the enzyme too that allows for the...
Correct. They produce the most of the enzyme that allows the shuffling. Yes. Because in our bodies, we do have variable regions in all of our cells, but they are highly regulated. So it's something where most of our bodies won't even produce the enzyme so that they can't shuffle.
Yeah, I just didn't know eukaryotes do transposons. I've never heard of that.
Yes, exactly. So we're very careful with what we create in the cells. So with this then, the take-home message is that with the same DNA, because we shuffle in the B cells, we make a unique individual cell each time when we're making a B cell. And so we randomly generate these antibodies before they ever come in contact with an antigen. So just like my drawing here, I have all these antibodies being released, and then their job is to find an antigen and see if they fit. If they fit, then that's great. So these antigens come in, our antibodies swoop down to see which one works, and whichever one works, that's the one that we clone. So we spend a lot of time making extra antibodies that will probably never be used. But our bodies do this as protection. They'd rather have all these variations present in case we need them than not have anything present and have us get exposed to something dangerous. So it's kind of a risk. It's an analyzed risk here. So one of the things we run into is that our antibodies are randomly made, and as we age, it is possible for the antibodies to end up with a sequence that targets a self-cell. So in other words, as I start to age, my shuffling process has been going on for many, many decades now. Now maybe I'm in my 40s, 50s, sometimes it starts in 30s. And what can happen then is I can get a shuffle where the antibody that's made is now actually targeting a protein that's on a self cell. We call this an autoimmune disease. An autoimmune disease is where our immune system gets confused. It's created an antibody against ourselves. Now, this can happen sometimes if we get, for example, a viral infection in an organ or in some tissue. The body will target the infectant, the antigen, and then it's producing a whole bunch of antibodies because it's on high alert, and it will have an antibody produced by mistake that starts to target the organ or the tissue. This is what's happened, for example, I told you guys I have thyroid issues. That's why I have Hashimoto's thyroiditis. I had a virus that got into my thyroid, and I produced antibodies for the virus, and now I have antibodies for my thyroid. So at the last count, I had 10,000 antibodies towards my thyroid. Not a good idea. We don't want to target my thyroid. It should be shaped like a butterfly. I have a half a butterfly right now. And he's slowly going away. He'll be a quarter soon. Because I'm old and my body got confused. Yeah, so that's why happened. I think. But it's because as things happen, we get confused. This is why, as we age, we're more prone to autoimmune issues. Now, sometimes this does happen early, though. We see sometimes younger people having issues with autoimmune disease. This is also what explains things like rheumatoid arthritis. They were born, and when they first started developing, they created antibodies mistakenly towards their bodies. So sometimes it happens right away. Lupus does this too. Sometimes it's something that's just a genetic defect, and that happens. but it's a mistake. Now, what's interesting is that in the body, they try to catch these cells that are harming our cells. So in other words, T cells and other B cells will try to regulate anything that they see that's attacking what it shouldn't be. So if a T cell sees this antibody binding here, it's going to try to actually attack it and take it out before it clones. It's like a race. Like the T cells kind of monitor. They're like seeing what's going on. And if they see somebody acting up, they're going to try to take it out. Sometimes, though, they just are able to clone before they can get taken out. And once they clone, they're there. And we can't get rid of them at that point. So again, this is what happens with things like lupus, rheumatoid arthritis, thyroiditis. A lot of times it's because we created antibodies incorrectly. So with this then, our B cells are going to be very important for protecting us against bacteria, for protecting us against viruses, and for protecting us against toxins. We're also going to have other cells that are going to help with our immune system, and that comes down to the T cells. The T cells, as I mentioned, can be either a couple of types. They can be the T lymphocytes, or they can be natural killer cells. So there's different types, and they do different jobs. They can look around and find pathogens and destroy them. They can help train B cells by showing them pathogens, so the B cells can try to attach and see if they fit the epitopes. They can also help detect self cells, so that means that they can attack anything that's not looking normal or self. That unfortunately could be implants, or that can also be any kind of cell that we have in the body that maybe is starting to look wrong, like a tumor. They are produced, remember, originally as a stem cell in the bone marrow, but they immediately get moved to the thymus. So they do not mature there. So they are produced as a stem cell, but when they move and make their journey to the thymus, that's when they become a true T cell. Okay, so the B cells stay, the T cells move out. Their job is that they recognize something called major histocompatibility complexes, MHCs. Woo, that's a mouthful. MHCs are basically the surface proteins on our cells. So this would be an MHC. And what they're doing is when they circulate, they bump into cells along the way, and they look for that secret handshake thing I told you about way back. They're making sure that the cells are self. If the cells don't respond, and if they don't recognize their proteins, the T cell is going to attack. It's going to mark them for destruction. It's going to call in other cells, and it's going to attack them. Okay? So it helps our bodies distinguish self from non-self. T cells do not make antibodies. That is all about the B cells. They can help train the antibodies, but they don't make them. All right? And the T cells, a lot of times, are also able to come in and help kill infected cells. If I have a cell that gets a virus inside of it, so a cell plus a virus, the cell sends out a distress signal, if it can. Now, this is a race between the virus and the cell. Remember I talked about how the virus uncoats and tries to get into the DNA right away, or the RNA of the cell? It's a race. Because if the cell detects that there's a virus there, it sends out an alarm signal, kind of like a burglar alarm or a car alarm. All the other cells nearby shut down. They stop bringing in outside nutrients, and then that way they don't risk bringing in a virus. So they all kind of shut down and wait. And a T cell will zoom up, and if it detects that there's a virus inside, it will inject or release lysozyme inside of it, and then that will encourage the cell to destroy itself. How cool is that? So these T cells, they're helping to target and destroy infected cells. So T cells a lot of times are also referred to as our helper cells because their job is to help the body stay safe. If something is detected as incorrect, they're going to help the cell destroy itself. They're going to walk it through its process of releasing its lysosome, or else they'll try to fuse with it to release its lysosome. They're going to also call in other white blood cells to help with phagocytosis. So they're going to mark it for destruction and be like, hey, neutrophil, get over here, eat this thing. You know, they're going to be like targeting it, telling them what to do. They also help our B cells recognize antigens. If they find a virus, the T cell actually grabs it and kind of shows it to the B cells. It takes it to the lymph node and it's like, see this? This is bad. You know, everybody see this? So it has like a training period. Our natural killer cells, they're able to destroy any cells that are present or contain any kind of microbes or any kind of surface proteins that they shouldn't. And let me explain that. When a cell gets infected, so let's pretend this is a cell, and let's pretend it gets a virus. Let me do a different color. Let's pretend it gets a virus inside. Okay, so now we have a virus in here. And remember, the first thing the virus does is it's going to uncoat and start doing biosynthesis. The cell is going to start having viral proteins present on its surface. That's one of the ways that the T cells can recognize that it's infected. It's a way that our cell can kind of call for help. And when it sees a foreign antigen on a surface of a cell, it's going to destroy it. Does that make sense? Because it can see basically what's happening inside. So if that's the case, then the T cell is going to mark that cell for destruction. And then this is fun. It also communicates with other cells in the area. It releases special proteins called cytokines that move in the cell's area and tell the other cells, hey, there's a virus present. Don't pull anything in. I'll let you know when the coast is clear. Again, it's kind of like if you have a police car on the scene and they say, you need to clear the quad for now. We'll tell you when it's safe. It's the same idea. That's what they're doing. They're like the little internal police. So cool. So with this then, those T lymphocytes are going to use that major histocompatibility complex, remember this, the surface proteins, to be able to recognize cells from cells that are either infected or there's something wrong with them. Now there's different types of MHCs. The first type or class is the major histocompatibility class that all of our cells have. This is the protein sequence that's found on the surface of all of our cells that say this is what I'm showing that I belong here. Okay? There's another set of cells though, the white blood cells, that are MHC2s. And these are going to be ones that are expressed when a white blood cell engulfs a pathogen. So remember I told you that if I have a cell that has a virus, it'll have these little particles on it, these little proteins that will target it for destruction. Our white blood cells, let's pretend this is a white blood cell now. Okay, it's a white blood cell. And instead of being infected with a virus, it's done phagocytosis. What's phagocytosis? Eating. It's eaten some cells. Let's say it's eaten some E. coli. So now it has some E. coli inside of it. Here's where it gets cool. The white blood cell who's eaten the E. coli, while it's working on destroying it, is temporarily going to show E. coli proteins. Okay? This is going to help show our B cells what E. coli proteins look like. It's a trainer. It's a learning size. The T cells are so smart, they recognize that this cell is a white blood cell, that it's actively destroying something, and so it won't target it for destruction. Does this make sense? There's a difference between a cell that's infected, a cell that's sick, versus a white blood cell that has eaten something, it's trapped it, and it's destroying or neutralizing it. But the beauty of this process, we need our cells to do this, our white blood cells to do this, because this is how we're going to train our antibodies, our other cells, how to recognize something that's dangerous. Isn't that cool? And your T cells are so smart. They know the difference between a regular cell showing proteins from an infector and a white blood cell showing up. So there's two different major histocompatibilities. There's the regular cells, which all have MHC1, which is their protein. If their protein sequence changes, the T cell is going to kill them because that means they're infected. There's MHC2s that are only found in white blood cells, and they are temporary protein markers that show that the white blood cell is killing something and it's showing the body what that antigen that it's killing look like. Does that make sense? Okay, good, because I know that can be complicated, but that's how the T cells are going to work. So this goes back and helps us with that idea of primary and secondary responses. So when we're first infected with something, those T cells are going to be helping to find cells that have been quote unquote infected, and they're going to be showing the B cells. So they're going to find white blood cells that are fighting and eating those antigens, and they're going to take that T cell and they're going to, the T cell is going to take that white blood cell and they're going to move with them into the lymph nodes, and they're going to call all the other B cells around. And those B cells are going to come in and they're going to see if they fit. And if they do, they're going to make a clone of them. Okay, it's a learning process. It's a way that our body is constantly building our learning. Yes?
If the body's reintroduced to E. coli, would the white blood cell just leave it alone because it already taught the T cell?
No, remember, they're non-specific. So the white blood cell is going to respond to E. coli, to staph, to Pseudomonas. It doesn't care. They don't have memory cells. White blood cells just attack. They're not our learning cells. But the antibodies are the ones that are our learning cells. Great question. I love that. So remember, white blood cells are all part of our innate immunity. So they see something, they attack. And that's why the T cell tries to work with them to say, wait, this is new. Let me get you some B cells because we got to learn what this is. So the T cells have a huge job to do. They're regulating, they're monitoring, they're teaching. They're like all encompassing here. Again, do you see why HIV is so insidious? They take out our T cell. We have B cells wandering around. They don't know what's going on. We have cells that are infected, calling for help, and nobody's responding. We have cells that are trying to learn, and nobody's there to guide them. So the T cells are so fundamental in our process of survival. Now, that first response then is all about learning. So when we're first exposed to any kind of antigen, we're learning. We're making antibodies. We're making clones. It's going to take us about 5 to 7 days to actually start to fight the infection. So that primary response, we're not doing anything. We're just learning. When we get to the secondary response, that's where we're now able to come in faster. We've got antibodies in place. Our soldiers are ready to go. They're trained. They know what they're attacking. And they're going to come in and try to limit that infection as soon as possible. So secondary responses are much better. This is also why we need booster shots every now and then. Booster shots are to remind us and to keep our secondary responses alive. Okay, so that way we remember what those attackers look like. Now, one of the ways that we can check and see how our antibody levels are doing is to do what's called a titer test. A titer test is looking for the amount of antibodies and the types of antibodies in the blood. And what we see is when we're first exposed to something, our primary response has a lower number of antibodies because we're learning. Then they increase. By the 5th to 7th day, I'll see their numbers shoot up. The secondary response, though, is much higher. So I have a whole bunch of antibodies in place, so my response is much faster. Now, Erin, you'd asked me a while ago which was better to use in blood testing, antigens or antibodies. And I told you there's really not a difference. And here's why. An antigen test is telling me whether or not, for example, COVID is present or whether or not flu is present. It's looking to see if the organism that's causing sickness is detectable in the blood. Okay? Now remember, antigen tests are just going to tell me that the organism is present. The antibody test tells me that I have created antibodies to fight that organism. The difference here, the antigen test is only going to be positive for a short period of time, while the antigen is actually present. Now with COVID, we get an antigen test that'll turn for up to 90 days. So we'll find traces, we'll find pieces of proteins for up to 90 days. So it can be a little false. The antibody test is like footsteps. It's looking to see what we've been exposed to in the past. And then we can do a titer test to see how many antibodies we have against the antigens at that time. Okay, does that make sense? Good. All right, so sometimes we run into issues though where our immune system has the best thought at heart, but it overreacts. We sometimes end up with hypersensitivity. And so this is where the immune system just kind of goes into overkill. And we see this, for example, with allergies. I told you, allergies are harmless, but for whatever reason, our immune system has decided they're deadly and they're going to save us from the pollen. It's like, I will save you from this evil pollen or whatever we're having an allergy to. I have an allergy to latex, it'll save me from latex. Latex isn't going to hurt me, but my body doesn't know that, right? So it gets confused. We also get autoimmunity, as I mentioned before. This is where we're targeting ourselves. So we've made antibodies that were incorrect. And now we're running into a problem where they're now going to be targeting us forever. We also get overreactions or hypersensitivity, where the immune system starts to freak out. That is what we see with the majority of our COVID patients that are hospitalized. If you check a COVID patient who is on a ventilator, you will rarely find a virus at a high enough level to even be detected. What that means is that their body's already destroyed the virus. They're not dying from the virus. They're dying from their immune response, the inflammation in the lungs, the extra fluid, all of the pressure that comes with that extra fluid, and the fact that they can't breathe and hold oxygen. That's what's killing them, their own autoimmune responses. So that's what we learned along the way, is they'll try to give them steroids, they'll try to do things to calm their immune systems down. But a lot of times it's too late, because once your immune system starts, it's in full gear. It's hard to stop it and turn it back around. So it's really fascinating how certain viruses can progress. So we also can run into immunodeficiency, and this is when we unfortunately have lost the ability to create our B cells or our T cells. Something's happened where we're having our antibodies and our T cells no longer being made correctly. When that occurs, then we can become immunocompromised. We have fewer T cells, fewer antibodies present, and that means that we now have a harder time responding or fighting off infection. So we are more prone to get ill. We are more prone to having illnesses every single time we get exposed. Now, sometimes immunodeficiency can occur through genetic issues. It can be something we're born with. Sometimes it can occur through mistakes made as we age, as I mentioned before, like with my thyroid. Sometimes it can occur through viruses, like with AIDS, or leukemia, like cancers. Something is affecting the immune system, and it's taking out our T's and B's. And so once we take this out, we're taking out first responders, second responders. We're taking out our police, so to speak. And so we no longer have anybody to help circulate and enforce and make sure that things are safe in the body. So let's just do a quick summary to make sure that all of this is making sense. Innate immunity is going to rely on immunity that is already present. So something that we're born with that's already present. It's going to exist from the time that we're born, and it's our first line of defense. It's also going to include our second line of defenses, which are our white blood cells. It has no memory. It is nonspecific, and it will react the same way every time an antigen is brought in. So whether it's the E. coli, whether it's the flu, they're going to react the same. They have no memory. They don't learn from each sequence. Adaptive immunity is different. Adaptive immunity is going to develop once we get exposed to an antigen or to a microbe. It is going to be a learned response. And your adaptive immunity doesn't usually kick in until our innate immunity has failed. We're going to build memory cells. Those are going to be our B cells that are going to remember what we've been exposed to. And they are going to help target that same antigen in the future. Now, what happens if a virus mutates? Are we in trouble? Yeah. If a virus mutates, that mutation, what that means is that the protein sequence that our antibodies once recognized have now changed. So when it comes in, we go right back to square one. We go right back to the beginning, where we have to learn what the antigen is. We go back to a 5 to 7 day wait period before we can even start fighting it. This is why mutations are so scary to us. We don't like mutations because it throws our immune systems off. That's one of the things we worry about. And remember, our T cells are going to be our protectors, that are what we call cell mediated. They're interacting with the cells. They're looking at those MHCs, those major histocompatibility complexes. They're looking to see, okay, is this a normal cell that has an MHC1, or is this a white blood cell with an MHC2? MHC2s mean that they're going to have sequences from the antigen that they've eaten and that they're fighting. The T cell will use them to help train the B cells, to show them what they look like and to target them in the future. So these are all part of our learning process. So primary immune responses, when we're first exposed to something, they tend to be a short-lasting effort to try to kill the antigen. We don't create a huge amount of antibodies, so they're smaller in magnitude, and we tend to have a longer learning period. Now, the second time we get exposed to something, we're going to have longer protection. So when I say longer in duration, I mean longer protection. We're going to create a bunch of antibodies. We're going to create more memory cells. We're going to remember this antigen. And the next time we encounter it, we're going to kill it fast before it can infect other cells. That's the goal out there. Again, the immune system sometimes, though, makes mistakes. It can become hypersensitive, where it's overreacting to things that aren't harmful, or it continues to overreact and doesn't stop. It gets into a positive feedback loop. I talked about that with COVID. We just keep getting more inflammation. We don't stop. Or we can become immunodeficient, meaning that we're not creating T cells, B cells, white blood cells correctly. All right? Okay, so that's the end of adaptive immunity. What do you guys think? Isn't this cool? Isn't it fun learning about how your body works? I love this stuff. Okay, so I'm going to stop here.